Hello, everyone. I'm Eric Wexler, VP of Business Development at CEDx Labs. Thank you for joining CEDx for our COVID-19 serological quantification webinar. It's my pleasure to welcome on behalf of CEDx Labs our two speakers, Dr. Aishwarya Venkataraman and Kevin Arnold, as well as all the attendees on the call. First, some housekeeping. Please submit any questions you have via the Zoom chat box. We'll have a Q&A after each speaker. Also, this meeting is being recorded via the Zoom chat box. So I will send you a YouTube link once it's uploaded. So as a quick introduction, CEDx Labs has been building a diagnostic menu for rare and emerging disease since 2018. Our expertise with ELISAs led us to support COVID-19 studies for the past year. Also, for the past several years, CEDx has worked with EUROIMMUN, and we are one of the few organizations in New England to have their EUROArray instrument. At this time, we are currently providing analysis using their dermatomycosis kit. Also, we do have their CLIA-validated Powassan test, and now their new EUROIMMUN SARS-CoV-2 QuantiVac serological testing kit. We also provide CLIA-validated lifestyle biomarkers, including thromboxane B2, a marker of systemic inflammation, and 8OH hydroxy to measure DNA degradation. Lastly, I'm also very pleased to announce that starting this month, CEDx Labs is the exclusive US and Canadian distributor for LifeLength, a Madrid-based company conducting a proprietary telomere analysis test used by doctors and organizations throughout the world to measure biological age by median telomere length. So I am honored to introduce our first speaker, Dr. Aishwarya Venkataraman, the Associate Director of Scientific Affairs at EUROIMMUN, a PerkinElmer company. Dr. Venkataraman holds a PhD in the field of neuroscience, and during her study, she identified novel autoantigens in autoimmune hyperexcitability disorders. She is also a designated inventor and holds a patent for the identification of a methodology for isolating an autoantibody binding to a polypeptide selected from the superfamily of proteins called soluble NSF attachment protein receptor in patients with select autoimmune neurological disorders. She has written multiple scientific articles regarding biomarkers and neurodegeneration, and her work has been published in high-impact journals such as Alzheimer's and Dementia. Now, since the beginning of the COVID-19 pandemic, EUROIMMUN has built an increasing important role in SARS-CoV-2 at EUROIMMUN US by providing both internal and external support. Her recent contributions includes the immunity count section of a Science article, and she was a subject matter expert for technology networks, COVID-19 antibody testing, S versus N protein. Ishu, let me see your screen. Wonderful. I can see your screen. Please start when you are ready.
Thank you, Eric. I hope you can hear me and also see my screen.
I can.
Awesome. Thank you. Thank you for the very nice introduction. It is a great opportunity to provide or give a webinar for CEDx Laboratories, whom we have been a partner for so long. Thank you once again for this opportunity, Kevin and Eric. So today I'm going to be talking a little bit about the immune response following SARS-CoV-2 infection. I guess this is a topic that many of you might be already familiar with, but also some of you might be interested to learn more. And that's kind of the goal today, to discuss in depth the humoral and cellular immune response following COVID-19 infection. So the presentation will take about approximately 25 to 30 minutes. And during this period, I will be going over briefly the company overview, that is EUROIMMUN, and then do a deep dive into the pandemic year 2020, and then talk very briefly about the immune response following SARS-CoV-2 infection, and then dive deeper into the B cell and T cell immune responses. I'll also be introducing the new EUROIMMUN QuantiVac assay, which is helpful for measuring the antibody responses following COVID-19 infection. And towards the end, I'll provide a short summary of my presentation. So to start who we are, EUROIMMUN is a medical diagnostic company. We were founded in 1987 by Professor Dr. Med Winfried Stoker. Until date, we have built over 3,500 products in our product portfolio in our catalog. In 2017, we were acquired by PerkinElmer, a Boston-based diagnostic company, for $1.3 billion. Today, we are a EUROIMMUN company, but also a PerkinElmer company. And by 2020, we have an annual revenue of about $450 million. We are proud to have over 350 patents in our catalog and over 16 different subsidiaries covering the globe. So what we do is we produce and provide state-of-the-art diagnostic solutions and assays for detection and diagnosis of autoimmune conditions, infectious diseases, including COVID, allergy, various genetic conditions, as well as we also produce various automation solutions so that our customers can seamlessly run all of our assays in a fully automated fashion. So to start with SARS-CoV-2 infection, I think we will go down in history as the year of the pandemic. The year 2020 will be remembered as the pandemic year. It is both exciting as well as unfortunate that we are all being a part of this experience. The unfortunate incidents, I think everybody is familiar, but the exciting experience is science did a lot of breakthrough advances just in 1 year. There's a lot of diagnostic tests available so that we can measure and diagnose COVID accurately in a very rapid pace now. And now pharma companies are working to release vaccinations in millions of counts so that they can help the world achieve the vaccination dose and ultimately reach the goal of herd immunity. Just to give you a snapshot, I think everybody is familiar, we've all been through this, but last year by March, I think the world went into a complete lockdown. And in the summer, I think every country was struggling and striving to flatten the curve. They're trying to help the people diagnose this infection accurately and as well as treat them. Towards the end of 2020, we had very good hopes because vaccinations were ready to be rolled out. We had 2 EUA approved vaccinations in the United States that were approved by the FDA. But also the world went into a little bit of more concern that there were new variants that started getting released. So because of these new variants, people and scientists had to understand further how this infection is going to spread and how vaccinations can help still prevent or be effective against these new variants. I think we are currently in this same world where a lot of us are getting vaccinated. I'm sure many of you in the audience are also vaccinated, but we are also in the concern that what does this variant mean to us? So let's talk a little bit about that. So overall, I'll start from the beginning. I think for SARS-CoV-2 infection, the biggest advantage is we wanted to fight this infection based on science. Based on science, I'm meaning the industry jumped in, developed diagnostic solutions for detection and diagnosis of SARS-CoV-2. And then there's pharma companies who is rolling out millions of vaccines to help provide those vaccination doses to people. But overall, it all stems from what is the virus actually doing once it enters your body. So very small, brief snapshot of that. So once a person is infected with the SARS-CoV-2 virus, there is the trigger of your innate immune response. Your body kicks in, and there are various immune cells in your body, that is like the dendritic cells, macrophages, B cells, that try to engulf this virus and alert the other immune cells in your body, which is then kicking the adaptive immune response in a high alert, meaning there is activation of T cells, which in turn activates the B cells, and B cells are the one which produce the so-called infamous antibodies. I think by now everybody is aware of what an antibody actually means. This is this is what COVID actually gave us as an advantage: more knowledge, more education to the people. So now you're measuring antibodies to understand your past infections. So then what happens is, after the initial periods of adaptive immunity is finishing up, there is then the body tries to remember this infection. There is generation of memory T cells and B cells, so that you can have long-term memory of this infection, and in case there is a reinfection, that your body still knows how to fight it. So it's just not the antibodies, but there is also B cells, there's also T cells, that is all fighting together in order to provide immunity or defend the body against COVID-19. So this timeline here just talks about what I briefly explained now. Just to reiterate, so once the body is infected with the virus, there is a surge in the viral load in the body. Immediately soon after that, there is the response, the immune response from the body that kicks in, the B cell, the T cell immune response, which lasts for a couple of months based on studies as till date. But years to come, how does the memory T cell and B cell protects us is something we still need to understand. So we'll be talking about a little bit about that as well in this presentation. So now to do a deep dive, specifically, how does the B cell immune response looks following SARS-CoV-2 infection? So like I said, initially when you have the viral infection, there is increase in the viral load, and this is the gold standard for you to get diagnosed if you have the COVID-19 infection. So there is RT-PCR methodologies, many of them EUA approved, and you can try to understand if you have a positive COVID-19 infection. So after that, a few days after that, between a week to about 14 days, there is the humoral immune response, part of the adaptive immune response that kicks in, meaning there is specific antibodies that are produced against specific areas of the virus. So there is initially IgA as well as IgM antibodies that is, rising in your body to fight this infection. So we have seen from publications that IgA antibody response is significantly higher compared to an IgM response, and an IgM response tends to wean faster than an IgA response. So both of them are considered an early marker of this past infection due to COVID. And then within a few days, there is seroconversion happening, and then there is production of very specific IgG antibodies against the coronavirus, SARS-CoV-2 virus. So again, this is the most crucial antibody because this lasts longer compared to the other isotypes. And we want to understand, based on this IgG response, how long do these antibodies last? How long do they provide immunity to you? Can they prevent reinfections, etc.? All of these questions are in everybody's heads and minds, and scientists are still trying to understand that portion of it. So overall, to understand all of this, you need test systems to measure what is happening in your body. So EUROIMMUN offers comprehensive and complete solutions, both for the diagnosis as well as detection, meaning monitoring through serology tests like the antibody measurement tests, etc. So on the left side of the slide, there is the molecular detection methods, the RT-PCR assays that are offered from EUROIMMUN, but I'm not going to cover too much into detail since today's topic is more about the serology test, or the test that can measure antibodies. So from EUROIMMUN, we have an ELISA system to measure the IgG antibodies specifically against the S1 domain of the spike protein. So this assay has been EUA approved since May of last year, May of 2020. And after that, since then, we have other assays that have also been released, which are research use only in the US, but to measure other antibodies in your biofluids like IgA, IgM, and against the other regions of the SARS-CoV-2 virus, like including the S1 domain, but also the nucleocapsid protein of the virus. But today, our focus of the presentation is going to be about the QuantiVac ELISA from EUROIMMUN. So this, I would say, is like a spin-off of our IgG ELISA against the S1 domain of the spike protein, but with calibrators so that you can measure the antibody concentration in the sample, right? So this is the EUROIMMUN QuantiVac assay, and let's discuss a little bit more about that. So last year, there was a huge interest. I mean, every single manufacturer, every single scientist, they wanted to understand whether there's an antibody response following SARS-CoV-2 infection. And at that time point, we had qualitative assays, our IgG S1 IgG antibody against the S1 domain of the spike protein is a qualitative ELISA, whether it tells you whether you have the antibody response, yes or no, kind of a result. But as we are progressing into this infection and as we understand more and more about SARS-CoV-2 virus and the infection that it causes, you want to understand a little bit more about, how long do these antibodies last? What is the concentration of these antibodies? And, what is the threshold above which it means that, you're protected or you're immune? And, long-term immunity, those are all the questions we want to understand. And hence, there is a need for measuring these antibodies in a quantitative manner in your blood. So for the sake of referencing it, it is a quantitative assay, but you have an antibody concentration. But in the US, it is addressed as a semi-quantitative assay based on recommendations from the FDA. So hereafter in the presentation, I will be addressing this assay as a semi-quantitative assay. So keeping that in mind, EUROIMMUN went and developed this assay, the semi-quantitative version of our IgG ELISA, because we know that it is already a high sensitive, highly specific assay, and we just want to measure the antibody concentration in them. So we included about 6 calibrators to this test so that you can measure the antibody levels. So this assay is also exactly similar to our previous EUA approved assay. It's measuring the IgG antibodies against the S1 subunit of the spike protein, which also includes the receptor binding domain. So what are the advantages of using such a semi-quantitative antibody response test? So as I mentioned, now you are in the era, you want to learn more. You want to understand more in terms of what happens after your infection, more in terms of what happens after vaccination. So you want to monitor your antibody response longitudinally post-infection or post-vaccination. So here again, the assay comes in to help. You can measure the antibody levels over a period of time and try to understand whether it's increasing or decreasing, or when it's decreasing. And overall, from the community, you want to understand long-term immunity. What are the levels of antibody that actually confer immunity to you, and how long do they last? We still don't know that. So, and then you want to do seroprevalence studies, epidemiological studies, and try to understand also herd immunity. So for all of this, you need to be able to numerically measure the antibody levels, and hence this is the advantage that this ELISA brings in the table, that it can quantitatively or semi-quantitative in the US, but numerically provide a value for the antibody levels in your sample. So now I just want to direct your attention to a little bit on the right side graph here. So from the beginning, EUROIMMUN decided to do an assay. Primarily, the first assay that we released was to measure antibodies against the S1 subunit of the spike protein. Why? This is just based on the experience from our R&D team, our experts back in EUROIMMUN Germany, who believed, based on their experience with SARS-CoV-1 as well as MERS, that the antibodies to spike protein is very crucial. Meaning these are the antibodies that ultimately will have the potential to neutralize the virus, potentially neutralizing antibodies. And ultimately, they also knew that this is the antibody that could last longer compared to other components or the other epitopes of the virus. And with over 1 year of us being in this pandemic, there's so many publications that has been released that talks exactly the same. So one such publication I've highlighted here. So in this study, they measured longitudinally for about 60 to 70 samples, and they found that amongst seropositive patients, so 50% of the seropositive patients became negative to antibodies against the N protein or the nucleocapsid protein in about 195 days. whereas 50% of the seropositive patients became negative to antibodies to the S protein against the spike protein in more than, after only 500 days. So this shows that, the antibodies to the spike protein last longer. And this is especially important when you're doing epidemiological studies, seroprevalence studies. You want to measure antibodies longitudinally, and you want to take a target that is helping you to measure them over longitudinal periods, and that is against the spike protein. So overall, the study concludes that the half-life for antibodies to N protein is approximately 50 days, whereas for the S protein or the receptor binding domain, it is more than 80 days. So hence, the use of measuring antibodies to spike protein is so crucial in this era. So now to talk a little bit more about the assay itself, some technical details. So like I said, there are 6 calibrators in this assay, which helps you plot the standard curve as shown on the right and ultimately measure the antibody levels. EUROIMMUN also provides the interpretation of your results, how to quantify them negative, borderline, or positive based on your values that you see following the analysis. And this assay, as I mentioned, is currently under review by the FDA for us. It's CE marked in Europe, so it's RUO at this time point in the US. But based on our internal studies and external studies, we have seen that the positive agreement to PCR, which is the gold standard, the RT-PCR, for greater than 15 days is about 93%. And the negative agreement to negative results, patients who were collected before the pandemic or those who were negative during the pandemic, the negative agreement was about 99.5%. So this is a pretty good, robust assay to measure reliably, quantify antibodies to the S protein. And then this assay has now been validated to measure the antibody levels in serum, plasma, and even in dried blood spots. So in case you are looking into a more easier matrix like dried blood spots, you can still use this particular assay. And this also is, can be fully automated using a couple of our automation solutions. So coming to the next part where, now we are having an assay, we are able to measure the antibodies, but the next thing, there are many manufacturers out there that are producing a similar assay. At this time point, you need to have something like a reference standard in order to harmonize or standardize the assay results so that you get similar results between various assays. So for that, the National Institute of Biological Standards and Controls from the WHO has released a reference standard, as I've shown here, for or to calibrate the anti-SARS-CoV-2 antibody diagnostic assays. So EUROIMMUN has already correlated our assay with this set of reference material, and we found excellent correlation with the NIBSC standards, R squared equals about 0.9. And based on that, you're now able to provide results in binding antibody units per milliliter. The value of interpretation I've mentioned here in the slides. So all you have to do is, once you have the results out of the assay in RU per mil, just multiply the results by a factor of 3.2 in order to report them as BAU per ml, which means that this assay has been calibrated to the NIBSC standards. So last year, using our qualitative ELISA, the same IgG against the S1 domain of the spike protein, we had seen many publications released by our external investigators that showed very good correlations between the IgG assay and neutralizing antibodies. So we expect nothing more, even for our QuantiVac assay, because it's exactly the same type of assay. It's kind of a spin-off. It's just a nice quantitative or a semi-quantitative measurement. And one such study is showing exactly what other studies have shown, a very good correlation between our QuantiVac assay and two neutralizing antibodies. So this study shows that the EUROIMMUN QuantiVac assay value above 49.8 BAU per mil predicted virus neutralization in about 95% of samples of dilutions greater than 1.5. So this just shows that our assay is able to detect potentially antibodies with neutralizing capacities. Additionally, this study also did a correlation to another surrogate assay, which also showed very good correlations to our QuantiVac assay. So overall, the study concludes the usefulness of quantitative assays in terms of determining vaccine efficacies, understanding seroprevalence or herd immunity in communities, and also understanding the thresholds of antibodies that need to confer immunity possibly, and how long do they last. So these are all the details that we are still trying to understand based on such studies, and such assays are crucial to help understand this data set. So internally, we've also validated our assay and correlated to the surrogate assay, which has been EUA approved for measuring neutralizing antibodies. And the data is here. We show about approximately 98% agreement between our QuantiVac assay and the surrogate neutralizing antibody assay. In turn, once again, emphasizing that this assay has the potential to measure antibodies or detect antibodies that potentially could be neutralizing. So our qualitative assay, which is EUA approved, recently was also approved for measuring antibodies to be included in COVID convalescent plasma donors. So since the QuantiVac assay is exactly similar to the qualitative assay, and the study that determined it also included our QuantiVac ELISA, we are hoping that this QuantiVac assay will soon follow the footstep of our qualitative assay to be included for COVID convalescent plasma. We're just waiting for the EUA approval. Once done, we're hoping this would be similar for the QuantiVac assay as well. Okay, so now coming to some important questions, which might be burning questions that the participants also might have. So you're having a QuantiVac ELISA, which is able to measure the antibodies, but when to measure antibodies, especially many of us are now that we are getting vaccinated. So as we know from Basic Immunology 101 and also from publications that has been released, it takes a little bit, 1 week to 2 weeks for the body to produce specific neutralizing antibodies. So therefore, keeping that in mind, we recommend measuring your antibody levels, especially if you're measuring post-vaccination, to measure them at least 14 days after your second vaccination dose. But if interested, if this is a study, you want to measure something earlier, we still recommend measuring 14 days at least after the first vaccination dose, so that you can get the full spectrum of antibodies in your body, which could potentially be neutralizing as well. And then, is it possible to differentiate between a natural infection and a vaccination-based immune response? So here, I'm going to specifically talk about the vaccines approved in the US. You have the Pfizer, Moderna, and the Johnson & Johnson vaccine. And all of these vaccines are based on the body to produce antibodies to the spike protein. The vaccines are mostly targeting the spike protein. So because of that, it is possible to differentiate when you want to, when a patient comes in and has just been vaccinated and has no prior infection to, SARS-CoV-2 virus, you can use the antibody like QuantiVac assay to measure the antibodies against the spike protein. And then you can have an assay to measure antibodies against the nucleocapsid protein. Because if the patient has previously never been exposed to SARS-CoV-2 virus, then there will not be any antibodies to the nucleocapsid protein. And hence, this will help you differentiate between the two in your studies. So this was actually done by a study in Germany, Dorschung et al., and they compared about 80 individuals who were vaccinated with the Pfizer vaccination, and they measured both the anti-spike antibodies as well as the anti-nucleocapsid antibodies, and they found majority or all of the patients positive for antibodies only to the spike protein. They just had one false positive result against the IgM antibodies against the nucleocapsid protein. But the author explains that it possibly could be that the person maybe has an early onset of the infection or got exposed to the SARS-CoV-2 virus recently before measurement. So, but overall, the study concludes that you can combine the assays, the anti-spike antibody assays like the QuantiVac and the antibodies against the nucleocapsid assay in order to measure immune response post-vaccination and differentiate them from natural infections. All right. So the next, I guess, burning question is the variants. Everybody is aware there is like various variants spreading out in the across the globe, like the South African one, the UK variant, etc. So how does this impact us, or how does this impact measuring antibodies via these assays? Long story short, it should not. Why? Because the body actually produces a polyclonal immune response once you have the vaccination injected in you. So it's not against just a single target or a single epitope, as we call it. It's against multiple epitope of the antigen, and hence the response is polyclonal. So the variants that we are seeing are right now point mutations, and hence just maybe one epitope could be affected, but your body still produces antibodies against all the other epitopes, which can still be detected by assays like the QuantiVac. So at this time point, it shouldn't be hindering, but internally we're working on studies where we are looking for samples with those classical variants and trying to do a study where with our assays to make sure that we are still being able to detect it. But of course, if the variants number of point mutations increase, if there is a structural functional change in the S1 domain or the spike protein itself, which ultimately means that the SARS-CoV-2 is completely changing its mechanism of a mode of action in the body, that means that we need to make sure that all diagnostic assays need to be able to measure the new variant, or the pharma companies need to be able to produce vaccines that targets those variants as well. At this time point, at this time point, the pharma companies are also mentioning that measuring their efficacies against the variants, there are varying results that are coming out, but we'll know more as more and more studies that come out in the near future. Okay, so next, I'm going to take a very brief moment to explain another type of immune response, part of the adaptive immune response, which happens, that is the cellular immune response following a SARS-CoV-2 infection. So if you remember the first graph, we talked about once the SARS-CoV-2 infection happens, maybe I can explain it here. Once you have the coronavirus in your infection, your body's innate immune response activates. You have various cells, natural killer cells, macrophages, etc., trying to signal other immune cells. Then your adaptive immune response kicks in. You have activation of T cells, CD4, CD8 T cells, which in turn then activates the B cells to produce antibodies. So you have the activation of T cells slightly earlier before your B cells gets activated or kickstarts. And all of these cells also produce an inflammatory cytokine called interferon gamma. Interferon gammas originally were known to be able to prevent or interfere with viral replication. So you can see how crucial it is. So, because interferon gamma ultimately can prevent the coronavirus from replicating in your body and prevent the spread of the infection. So hence, we feel that the T cell immune response, again, this is a very arbitrary graph, T cell immune response could happen either very close to or simultaneously together with the B cell immune response in the body. And it's crucial to also measure the cellular immune response generated by the T cells. So there's already many publications that are being released that show the importance of cellular immunity in COVID-19. And this one publication I want to direct your attention to says that specific T cell immunity or T cell response has been detected up to like about 6 months following COVID-19 infection. And it just shows how important T cell immune response or cellular immune response is also important for fighting this infection. So on what we know till now, we understand or we know from publications that about 20% of patients who recovered from infections were without any detectable antibody titers. So these patients recovered, but they didn't have any antibodies in them. So here we think it's possibly because of their cellular immune response that kicked in. So it's important to measure those immune responses here. And you have asymptomatic patients, some percentage, and also some percentage of symptomatic patients who become negative for antibodies within a very short time frame, like 8 weeks. And here again, you need to make sure that they are still protected or they're still fighting against this infection. And here is another opportunity to measure cellular immune response. Again, there are a lot of vaccination studies also measuring their efficacy based on the cellular immune response, making sure that this is also elevated post-vaccination. And based on recent publication, this immunity or immune response can be detected up to about 6 months following an infection. But what we need to know, or what we need to emphasize here, is that both the measuring the antibodies, the measurement of antibodies against the spike protein, the levels or the threshold, or the levels of cellular immune response in the body following vaccination or infection, how does it correlate to immunity? Meaning, is there a value that shows that if you have this value means you're protected? No, we don't know that. That is why these assays are available, so that you continue to measure your patients following post-vaccination, post-infection, continue to do studies. So more and more research is what will tell you that response in terms of what value of antibodies are needed, or how much cellular immune response is needed in order to obtain that immunity that you're looking for to prevent reinfections. So currently we are still in that unknown phase, but we hope this year we'll come to have some new information on that as well. Very briefly, the assay for measuring the IGRA, as we call it, the interferon gamma immune response following the infection, COVID-19. So what you got to do is just stimulate the patient's blood or the sample with the specific antigen. Here, again, we use the spike protein, S1 domain. And once this is stimulated, you start having the activation of T cells, which in turn produce the interferon gamma. And the interferon gamma that is released is measured using the EUROIMMUN ELISA, specifically to measure these interferon gammas. And this is then reported in units that's measurable, so that you can quantify or semi-quantify the level of cellular immune response in the sample. At this time point, this is also a research use only assay in the United States. So with that, I come to the end of my presentation. So I think we understood a little bit about the role that humoral as well as cellular immune response plays following SARS-CoV-2 infection as well as vaccination, and how important or how crucial it is to measure both these type of responses, because we are in the phase where we fully don't understand the virus as yet. The virus is also mutating, so we need to have constant measurement data points so that we can understand the infection even more, or the virus even more, how they are trying to, spread infections. So hence there is the need for measuring these assays, semi-quantitatively in the US, both following humoral immune response as well as the cellular immune response needs to be measured following a natural infection as well as vaccination. At this time point, we don't know the threshold or what are the levels of antibodies or the IGRA, that is the interferon gamma, that is needed to provide immunity. We are still trying to understand that. And EUROIMMUN QuantiVac, again, is an assay that can semi-quantitatively measure IgG antibodies against the S1 subunit of the spike protein. And this assay you can use with serum plasma as well as DBS samples. So with that, I just wanted to also provide here another information. We generally at EUROIMMUN, we host also series of webinars, and our next webinar is on nail fungal infections, infections caused due to dermatophytes, as we call dermatomycosis. And this is the assay that CEDx Laboratories has also implemented and is rolling out. This webinar is going to be hosted on June 3rd. And we hope you can also join us during this webinar. We'll be sending out invites very shortly. So with that, I would like to thank everybody for your attention and participation in this webinar. Thank you.
Well, thank you very much, Ishu. Very interesting. We do have some questions. The first question is, why is QuantiVac negative results still have an antibody amount of lower than 8 RU per mil? Are you able to address that?
So yes, so you want a result. It doesn't mean that just because you have 1 RU per mil or 2 RU per mil, it means that you're specifically having those specific antibodies against the SARS-CoV-2 virus. You sometimes have some level of immune response, possibly due to molecular memory, similar epitopes happening due to other infections in your body. So hence, we always determine assay performance. So with that, our positive percentage to agreement to PCR is about 93%. Its assays are always not 100% sensitive or specific. So it's about 93%, and it's about 99 0.5% specific. So to allow for that, meaning to detect only the specific antibodies, that is showing that this is a specific to SARS-CoV-2 virus, and it means that this is a virus that is caused because of the infection or the vaccination, and it is going to possibly rise. Because of that, you need that level of threshold of negatives, and above which is what is your specific immune response to SARS-CoV-2 virus. So does that answer the question?
Thank you. Another question is, what is the correlation between the identification of IgG antibodies to spike RBD and neutralizing antibodies?
So what is the correlation between IgG antibodies to spike, RBD, and neutralizing antibodies? Correct, Eric?
Yes.
Okay, absolutely. So maybe I might have to go back to some of my slides here so that I can show you. So there are many papers that I've released that show excellent correlation to antibodies, IgG antibodies against the spike protein as well as RBD protein. So one, we need to understand that the spike protein is where it hosts the RBD. The receptor binding domain is on the spike protein. So when we take the S1 subunit, it already has the receptor binding domain. You're still measuring antibodies to the whole protein itself, which includes receptor binding domain. But there are some assays which take only the portion of receptor binding domain. But nevertheless, it's a part of already the spike, spike subunit. This is spike protein. So we have shown excellent correlations between the antibodies to RBD, antibodies to spike protein, maybe some level of differences, I would say not too much. And, just to go back to this presentation, here they measured antibodies to spike as well as antibodies to RBD. In this particular study, I think they are showing here the percentage of seropositivity for about 195 days, where 50% of them become negative for the nucleocapsid protein. Whereas for the RBD, they have a slightly lower number. It's about 260 days. And for the S antibodies to the S protein is more than 500 days. So in this study, they show slightly lower longevity of antibodies to RBD compared to the spike protein. But there are a lot of other studies which show more or less similar longevities, and that's why excellent correlation. And to answer the second half of the question, again, so antibodies correlation between our IgG, again, QuantiVac assay and neutralizing antibodies, they are also excellent. So this is one study. They show excellent correlation between our assay and microneutralization test, which is like another version to determine neutralizing antibody. And here they've given an optimum cutoff to determine optimum values of neutralizing antibodies in your sample. So here again, they show very good correlations. And we have done an internal study, which between our quantitative assay and a surrogate neutralization test, which is not the PRNT test, it's a surrogate test. It's similar to an ELISA, and this is EUA approved in the US, so we did a correlation there. And here again, we see very good correlation between our assay and the surrogate test, and the agreement is about 98%. So very good correlation in term, meaning we are able to detect antibodies which could be potentially neutralizing.
Thank you. We're going to address a couple more questions, and then any question that hasn't been covered, I will still pass to Ishu, and we'll be able to answer your question in a more direct fashion. So the last two questions are going to be, what is the limit of detection for the test? After what value do you say is greater than max? Has WHO set a limit for detection for the BAU?
Mm-hmm. Yep, absolutely. So I'm going back to this slide where we have done the correlation with the National Institute of Biological Standards and Controls. So in our assay, the highest calibrator level is 120 RU per mil. And the WHO is in a similar lines as well. It's quite similar. I don't remember the exact value, but it's very close to that. Right now, to achieve a linear correlation, this is the standards which we can take. So let me go back to here. So this is the standard curve or the calibrator value we take. It starts from 1 RU per mil to 120 RU per mil, and this correlates very well to the WHO standards. Beyond this, there is some level of linearity offsets that happen, which was done by our R&D team, and hence we don't want to go beyond this calibrator. At this time point, this is reported as greater than 120. And if you see that level of antibodies in your sample, mainly because of vaccinations, we are seeing that. So we recommend further dilutions. We recommend dilutions of 1:1010 or 1:100,010. Again, we'll be happy to provide you those protocols. So in order to get your sample value into the reporting, into the calibration curve, and then you would multiply with the factor that you diluted and then get a final value. But again, happy to help the participants in case they want any specific protocols. But right now, our maximum calibrator value is 120 RU per mil.
Thank you. And the last question I'll deal with on this webinar is, have you seen a difference in IgG levels between the vaccines, particular demographics, i.e. sex, age, or BMI, or reaction to the vaccine?
Again, we haven't done many studies. There are many studies undergoing because of vaccines. As manufacturers, we are trying to make sure that how our assay reacts post-vaccination, but we have not done extensive studies on vaccines and how it reacts or the vaccine efficacy changes based on demographics. So unfortunately, I'm not sure if I would be the best person to answer that question.
That's where these upcoming studies, once this has EUA approval, hopefully we'll be doing a lot of work to be able to answer that.
Yes, yeah. And many pharma companies are also doing many such studies. A lot of publications have been out there. They are including various demographics of patients just to make sure their efficacy is remaining stable. So such publications will also be helpful to read.
Well, thank you very much, Ishu. It was a real pleasure. Now I'd like to introduce my colleague and one of CEDx Lab's co-founders, Dr. Kevin Arnold. Kevin recently left Bioagilitics after a 4-year role that spanned both CRO as well as the diagnostic business units. He effectively managed day-to-day operations from sample processing to providing patient results. His significant contribution supported Bioagilitics' acquisition of Cambridge Biomedical in 2019. Kevin's expertise includes strategic operations development, identifying esoteric testing growth, consulting on best-in-class platforms that CRO clients need, including cloud-based EDMS, disaster recovery, and next-gen LIMS capabilities. Before Cambridge Biomedical, he was Director of Lab Operations and Genetics at ReaperSource Fertility Diagnostics. Kevin earned his PhD at the University of Aberdeen, focusing on molecular parasitology, and his BS from Bangor University in applied zoology and marine biology. Kevin, would you like to share your screen?
Yeah, so hopefully everybody can hear me. Right.
We can hear you.
Okay, can you see that is the question?
And we can now see your screen. So you are good to go. Please begin when you are ready.
Thanks, Eric. Wonderful talk issue. Mine's going to be a lot shorter and a lot less slick, I'm afraid. I'm not the professional speaker. So CEDx Labs, why did we basically, this is going to be a why did we choose the EUROIMMUN kit, and why do we think we should be testing this in the IgG serology? There we go, exactly, topics. Why did we choose EUROIMMUN's anti-SARS-CoV-2 QuantiVac ELISA IgG? Our validation experience, very limited, and the potential and importance for this testing in the near to mid-term. That's the big one for us. Why we chose the EUROIMMUN? I mean, Issue's made a very solid case for it. Yes, it's semi-quantitative, and obviously we would love fully quantitative, but there is no internationally approved standard for the quantification of the IgG against SARS right now. There's a working standard, which EUROIMMUN did a lovely job demonstrating good correlation. And so our hope for the future is that, eventually that standard gets to be fully approved and everybody can go quantitative against that. But the semi-quantitative is as good as we can go right now. And the nice thing is, as you said, this is a build-off of the qualitative version that did get EUA approval. And even though the FDA has definitely slowed down in the EUA acceptances or EUA approvals, they're still pushing out a few at least. So we're hopeful that the FDA EUA comes through for the QuantiVac test. Solid test performance characteristics, the qualitative versions are already out there, so that's great. Cross-reactivity and interference, EUROIMMUN did a nice job on that. It's very hard for small labs like us to really do the cross-reactivity stuff in any useful form. Wide calibrated, 0 to 120 relative units per mil, potential to quantify very high values. This is a big one for us because the initial sample dilution is 1 to 101, which translates to 10 microliters to a mil. But the post-vaccination samples that we've tested to date, they've been way up there. So we've actually had to do higher sample dilutions than that. We're not going anywhere as high as issue was suggesting. We were doing like 1 in 500 basically. Relatively short. This is an operational one. It's a very relatively short assay, 2 hours of incubation time, prep, washing, reading, approximately 3 hours. So you can do a couple of batches a day, which is great. Relatively low defined positive threshold. The question's already been asked. What does positive mean? And in our belief is it's going to be post-vaccination. So it's yes, you're going to be positive based on this assay for hopefully a very long time. But your numbers could, it's effectively going to be the tracking of the drop of your numbers from your high after, a couple of weeks after that final vaccination. So our verification, we just did inter-assay precision, accuracy, and linearity. Nice simple ELISA to run. Nothing too special. Our precision was a bit worse than the EUROIMMUN's, but we still, less than 20% CV. We crossed a range of stuff, 98, 23, and 4. Our accuracy back currently from calibrators is very tight, but we're going to redo that with the NIBSC material anyway. Solid linearity, yeah, we generated a nice linear using the serial 8.0 dilution. And NIBSC also now has this nice QC reagent, which is this 20B764. And this actually has lot number specific EUROIMMUN values. It's got a bunch of other tests in there as well, but it's got lot specific EUROIMMUN values. So that's going to be a great QC to run, routinely when you're doing the testing. This is the crux of the stuff. So why test? Well, we believe, post-vaccination environment, at least in the US, you'd like to believe that everybody's going to get vaccinated. You can make a test for, make a case for testing, exposure, asymptomatic cases, but realistically, nobody cares, right? If you're an asymptomatic, you're an asymptomatic. You have no symptoms. Why do I care what your antibody levels are at that point? The initial vaccination thing, at least in the US, except for J&J, which is now on pause, it's 2 doses for a vaccine. Do people care after that first? I get my first dose, my first shot. 2 weeks later, am I interested in what my antibody response that I generated is? Now, certain people will be interested, but realistically, is that going to stop them going and getting the second dose? It shouldn't. And who's going to be paying for your serology testing? Halfway through your vaccination regime. That's a pretty limited ticket. The big one is obviously longitudinal analysis. Am I still covered? So, knowns, unknowns, cliche, totally, but it's totally legit in this case. There's way too much that we don't know. And this one is obviously the big one. Am I still covered? Do I get vaccinated? How long is that good for? There's going to be no simple answer, as people say. BMI, gender, age, everything's going to play in there. So grayscale at best, in my opinion. Somebody's likely to come up with a singular threshold value. But how good is that value, and who's coming up with that value, and effectively, what does it mean? However, this is literally this is the first pandemic in my lifetime, and probably in most people's lifetimes, and where testing will be a key component of getting back to the pre-pandemic normals. Large-scale use of this testing over time, diverse demographics. This is a data set that we just don't have this. And I mean, yes, it's a horrible scenario, but this is the data set you really need. You're gonna now vaccinate a very large proportion of the population with a disease that they haven't seen before, and is it going to be like flu? Are you going to be trying to do it every 6 months? Are you going to be trying to do it every year? Is it going to be bundled in with the flu vaccines? How long does the immunity last, or is it just going to sort of drop away to nothing and disappear away? You don't have the data sets to do any of this right now. There's been a narrow window. The 2 vaccines came out in December, and you're trying to vaccinate the entire US by hopefully the end of May, shall we say? That's a very, very narrow window for everybody got vaccinated. Okay, so does that mean everybody, 6 months, 12 months from now is going to need a second set? Logistic and lifestyle calculations. So you need data-driven, robust answers to the when do I need another shot, and what and who is defining those cutoffs. Monitoring of high-risk environments. This is where we think it's going to be. We think there's a nice business in this piece. These are environments where people don't move around that much. Nursing homes, prisons, hospitals. Hospitals is an outlier, obviously, because they're clinically based, and infection is their business. The nursing homes and the prisons, the early data from the prisons was primarily asymptomatic. So it was pretty opposite from the nursing homes. But the folks in the nursing homes, they got first doses, but they're also in the age group where vaccinations are not going to give you the most robust responses. So that will be a population that we think should be monitored pretty routinely to get yourself hey, we think now's a good time that all of this nursing home should now all get revaccinated. Vaccine comparisons, another data set that you have for no other disease. There's more than four vaccines out there. Obviously, I haven't got the Russian vaccine out here, and I haven't got the Chinese vaccine out here. But having said that, Pfizer and Moderna are US, AstraZeneca is Europe, J&J is now effectively both. But there's also been, within and between the countries, there's been very different strategies. All right, Pfizer and Moderna in the US, 4 weeks between doses, and that's the game. UK, they've done everybody gets 1 dose, and then 12 weeks later, that's the second dose. But they're also primarily AstraZeneca. So that's going to be a data set that sort of, the farmer should be certainly interested in that. And purely from the epidemiology point of view as to was my strategy the right one, at the country level, that's also going to be a lovely data set. And then the rules for pre-pandemic activities. What's legit for going back to school? What's legit for going back to work? And events, high people density activities. When can I go back to Fenway Park? When can I go, go watch U2 concerts? When can I go and sit in the theater again? This is also more of an ethical question as well, because it's gets to the vaccine passports type stuff. Obviously, international travel for us is right now there is no international travel without PCR, basically. But there's been a lot of talk about vaccine passports, and people have been talking about invasion of privacy and all that. But yellow fever vaccination status has already been required for travel to certain African countries for a very long time. So this is not necessarily that different. I mean, WHO recently just rejected the vaccine passports, but the rationale for a rejection was just the inequity of vaccine availability. There's a whole chunk of the planet right now that doesn't have anywhere near the same level of access to getting a vaccine, hence the rejection of vaccine passports. However, the African CDC then put out the Common Continental COVID-19 digital passport, where hey, you have the you have this digital passport, you can travel anywhere across continental Africa. EU's going the same way. They're trying to release the digital green certificate in June for, hey, my vaccination status, here you go, nice little digital setup, or, through like trust and stuff like that. But what's okay in 2021? I mean, this is the US right now. This is an executive order out of Florida, like 2 or 3 weeks ago, prohibiting COVID-19 vaccine passports. And it pretty much sort of just said, hey, you can't force me to prove my vaccine status for me to go about my regular life. That's just a statement from the governor. And how are good for how long? That's the big question. It's just like right now, the CDC FAQs basically says, this is the statement from them. How long does protection from a COVID vaccine last? We don't know how long protection lasts for those who are vaccinated. And then, New York Magazine, how long does immunity from COVID vaccine last? Right now, that's how long's a piece of string, and it's going to be different. And unless you start measuring serology longitudinally and come up with hey, we believe that this value still means you have decent level of immunity to COVID infection. There is no data, right? I mean, there's they've got 6 months of Pfizer just put out their 6 month stuff saying we still look good, but this is a huge number of people have just been vaccinated. This is going to be the hey, how long is it? When do I need to get a new shot? That was it, really.
Oh, thank you very much, Kevin. Excellent. It gives a very nice overview of the different issues that are facing the usefulness of the serological testing. So we do have a couple questions. I do know that this is scheduled to noon. We have this for another 10 or so minutes. So I'd like to at least follow up with a couple of these. And again, if you do have to leave, thank you for attending. I'll be sending out an email with links to both talks when they're posted on YouTube. Feel free to reach out to me to discuss any way CEDx Labs may be able to support your diagnostic and bioanalysis needs. And Kevin, one question I'm punting to you is, how can we implement antibody tests to results to create a safer work environment? You sort of skirted how that is going to be important, but what type of strategy might you suggest?
That's come up locally, actually, to be honest with you, because I mean, anecdotally, shall we say, employers can't ask for your vaccination status, and so what I'd recently heard was, we're all a family. You can come back to work so long as you've been vaccinated, but we can't legally ask you. This is the US, obviously. We can't legally ask you what your vaccination status is. I mean, that's a difficult one, right? I mean, schools, universities, they have I think you can make currently you have to have certain vaccinations to be able to go to universities, to be able to go to certain schools. The workplace environment, I don't think I would have believed that a private company could actually either mandate that you prove your vaccination status, or if you don't want to have the vaccine, you're offer that you can still work from home, all that sort of stuff. Right? I don't legally, I don't know what the options are for doing serology testing at work. Now, obviously, I think you could offer it, but I don't think you could actually mandate it.
Yeah, let's just say it's a developing legal headache.
Certainly in the US, definitely.
Yeah, there's one technical question. I think we maybe all cover issue if you're still able to unmute yourself. We got, why is the S domain chosen over the whole S protein? Vaccines are designed to make whole S protein in the organism. Are you able to address that?
Sure. The reason why we went for a more specific domain is, long story short, is to prevent cross-reactivities. We want to make an antigen, we want to have antibodies against this antigen, which is highly specific to the SARS-CoV-2 virus. We have the MERS coronavirus, we have the SARS-CoV-1 virus. And when there is analysis done between the whole S protein, the N protein, there is a large level of homologies between all the coronavirus families, right? So we want to take a epitope or a region of that protein, which is more specific to the SARS-CoV-2 virus. And hence you're detecting antibodies to a specifically to the SARS-CoV-2 virus, in turn reducing cross-reactivity possibly to the other common coronaviruses and maybe SARS-CoV-1 and MERS. So this is one of the reasons. And we also knew that S1 domain includes also the R receptor binding domain. So that way we are potentially, we will be able to detect antibodies that potentially have neutralizing abilities.
Great. I knew you'd be able to comment on that. And then I think this will be the last question. What's more important, levels or the antibody avidity? Are you able to guess what may be more important, overall antibody levels or the avidity of the antibody?
Eric, is this for me or Kevin?
This is for you, Ishu.
Oh,
I
okay.
think you're.
Got it. Okay, so this is a big topic. So what is more important, antibody levels or avidity? I think we need to know a little bit more. There are already many viral infections where you want to measure avidity, making sure that it is a more recent infection, not a past infection. And we are not at this stage. We are just 1 year into this particular infection, and we are now measuring antibody levels. We are trying to possibly quantify antibody levels, try to get a number associated with it. As we progress into more and more understanding the virus and more understanding the immune response that the virus elicits, you then will start measuring, avidity levels as well. Meaning you want to make sure the antibodies that you're detecting are more specific for the more recent infection, specifically for IgM antibodies, more than the others, and not like a past infection. This is more or less, actually, we have assays to detect avidity response in especially those infections that you want to monitor during pregnancy, the TARCH infections. But I feel that we are now in a very infancy state. I think as we develop more or we understand more about this infection, we'll have a better understanding of whether to measure just antibody levels or avidity. Right now, we're in the phase of measuring antibody levels, if I can put it that way.
And then the last question, I think this also goes to you, Ishu, is does the correlation show agreement with PRNT?
Correlation shows agreement with PRNT, yes. So we have a lot of studies that have been released, or peer-reviewed journals that has been released, that show very good correlation between our IgG qualitative assay, the previous EUA approved one, against, again, the same S1 subunit, and PRNT tests. So we show a very good correlations, 0.9 and above, for measuring these IgG antibodies. Right now, there is a lot of ongoing studies. I think a couple of studies soon will be releasing publications that show good correlations between our QuantiVac assay as well as the PRNT test. So the one that I showed you in this presentation was against a microneutralization test, which is also an approved test for measuring neutralizing antibodies.
Well, thank you very much, Ishu and Kevin. We've gotten some great feedback. I'll make sure to share with you. They love your talks, and everyone, we appreciate you joining us. Be a lookout for a press release about CEDx Labs being the exclusive distributor of LifeLink's telomere analysis technology. So take care, stay safe, and have a great rest of your day.
Thank you, everyone.